Please use the link below to get the notes and other videos.
Again, we have started a chapter of doing questions and answers, and then if you have any question, maybe you have a paper you want us to go through, you can send that paper to us, and then we can go through that paper, as long as it is known, number one, SBA, because SBA we cannot go through SBA, but if it is something which is similar to it, then we can go through it, and then maybe give you ideas. SBA can only be done if it's a teacher who is asking for help. It's fine, you can send them to us. Any question paper you want us to go through, then we shall go through it. If you want the specific sections, then also we can also help you in that regard. Don't forget to subscribe to our YouTube channel, and then also to like, and also to send a link to others so that can also benefit. Don't forget that our channel is physical science, geography, life science, and then even others will be coming, but we have started with grade 12, and then other grades will be coming. Only physical science and the life science is now with grade 10, but even other grades, other subjects, they are going to go to grade 11 and also grade 10. Grade 9, we are coming for you also. All right, the question is saying that various, in this case, please, let's talk about some of the mistakes students do under objective. When we ask you to answer, they're asking you to answer A to D. So answer is either A, B, C, or D. So when you are trying to answer such question, don't rewrite the question. Some students write the question. Number two, when the answer is A, for example, don't write A and then you write Darwin and then Lamarck. No, we only want, the question is asking you A, B, C, or D. That's it. And then now once you write others, they don't even write A, they are writing the names of the, that's wrong, completely wrong. It means that you have not followed what? The instruction. Number two, questions regarding to objectives, you cannot leave it unattended. Why? Even you can guess, you never know that your guess can be correct, and then you get a mark. You cannot leave a question which is an objective unattended. Even if they're saying that there is only 1 second remaining, just circle or just write A, you never know. You can get those 2 marks from that. Yeah, so you cannot leave an objective unattended. If I see a student, for example, on my case, that has not attended an objective, I become furious. Why? Because that is a question. If you do have not understood it, at least just write an objective, any letter which is between A and D. You never know, you can get it correct. All right, let's start with the DNA. The structure of DNA molecule that was discovered by, the structure of DNA molecule was discovered by. So they have given you the name of scientists, the name of people who contributed to science based on our grade. So when we talk about DNA, remember there are 4 people who are involved. There are a lot of people who are involved in DNA, but based on our examination guideline, it's only 4 people: James Watson, Francis Crick, Maurice Wilkin, and then Rosalind Franklin. So you have to know that James Watson, they were together, and then Maurice Wilkin, and Rosalind Franklin, they were also together. You understand? James Watson and Francis Crick, they brought up the final DNA structure. And then Maurice Wilkin and Rosalind Franklin, yes, they're also together. And then they brought the initial stage of this DNA discovery. If you don't know, just look, check on our YouTube channel. The video for that is there. Or if you don't know it, you have failed to find it, just send us a message, and then we will give you the what? We will give you the link for that. All right. So they're saying Darwin is not involved in DNA. Lamarck is not involved in DNA. These people are involved in evolution. So we're going to see these people where? We're going to see them where? In evolution. All right. So we see that Darwin and Lamarck, these ones are involved in evolution. So we don't see them in DNA. And what about Mendel? Mendel is involved in genetics. We see that Mendel is in genetics. Watson, yes, we see that Watson is there, but Watson is not with Mendel. So we can't give it a correct answer. And then what about Franklin and Wilkin? Yes, these people, yes, were involved in the DNA discovery. These two people, ne? But this Wilkin, and then Franklin, they didn't discover the structure. They found out the initial stage of this discovery of the structure using the X-ray diffraction pictures, and then they took it to James Watson. So James Watson and Francis Crick are the one who tried to find out the correct answer. So the answer is going to be, we're going to find out that the answer is D. Then we go to the structure of, we don't know which structure is it. This we might say that this is DNA, but may not be DNA. Let's find it, first analyze it properly. They said this is molecule 1, this is molecule 2. Yes, it means that these are different molecules. If you look at this, you find thymine. So you remember that thymine is only found in DNA, while this one is having uracil, and uracil is only found in RNA. So it means that structure A, molecule A is DNA, while molecule B is RNA. You see? So it means that now, yeah, this is what is the difference. So now, since you have got that information, now you can say an observable difference between molecule 1 and 2, an observable difference. Molecule 1 is double-stranded. Molecule 1 is DNA. We understand that molecule 1 is DNA, but it's not double-stranded here because they said observable. They want it, they want what you call observable. So if it is not observable, you can't see it here. So we can't classify it to be double-stranded. Then they are saying that molecule 1 is double-stranded, while molecule is single-stranded. Molecule 2 is single-stranded, we see it, but molecule 1 is not double-stranded, so it's not a difference there. Then they're saying molecule 1 contains deoxyribose sugar, and then molecule 2 contains ribose sugar. It contains deoxyribose sugar, and then this one contains ribose. But how do you identify it? You can't see it with, because they said observable. Then number 2, molecule 1 has thymine, while molecule 2 has uracil. Yes, you can see it by your eyes. Is this one is thymine, while this one is uracil, and then molecule 1 is longer, and molecule, they are two, the two molecules are equal. So the answer will become, we will take answer C as the most correct. Yes, as the most correct answer. Then we go to another question. The same diagram below shows the DNA profile. Yes, remember DNA profile is a technique. DNA profiling is a technique used to identify someone's DNA profile. It means that this person is unknown, but we have to compare it with a known one using a known DNA profile. The definition was removed from the examination guideline, but at least you have to know it before you go, because you can't deal with something which you don't know. So that's why we have to keep on giving you the definition so that you know exactly what is happening. Yes. So now they're saying that now you have, remember, you have to be with a ruler and then you draw so that you see which one is matching. So they're saying that the parent of Zintle and the Ayanda have 4 children, and then the 2 children are their biological offsprings, while the other 2 are not biological, meaning that they are adopted. So if they are 2 and 2, let's find out. The mother and the father, their DNA must match with the children. Okay, let's start. They're saying which of the 2 are adopted children of Zintle and Ayanda. So now we say draw. This one matches, but this one matches from the father. Then nothing, nothing. This one matches with the father. And then this one matches from the mother. This one matches from the father. This one matches from the mother. So this one is covered, meaning that this one is a biological child. Then we go to Gugu. Gugu, nothing, 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 nothing. So Gugu is not. So Gugu is not a biological child. Then you go to Bandile. Bandile, this nothing, nothing. This one matches from the Zintle. Yes, and then this one also matches. Also, this is matches from Zintle. Again, this one matches from the father, and then this one matches from the father again, and then this one matches from the mother. So it means that Bandile is also a biological child. Then what about Li Zui? Li Zui. Li Zui is nothing matching. It matches 1, matches 2, nothing matching here. This one matches, but not all of it. It means that this one is not a biological child. So it means that Gugu and this one, number 1, Gugu, and this one are the adopted children of these 2 parents. So this one and this one, because they match with their parent, therefore we say that those are biological kids. So the answer is going to be the Bandi, Gugu, and so the answer is going to be B. Then they're saying that DNA molecule code for a protein containing 150 amino acids. Remember, we call it a protein. We call it a protein if it has 50 amino acids, amino acids and above. If it doesn't have 50 amino acids, then we don't call it a protein. We call it a polypeptide chain. We call it a what? A polypeptide chain. So that's how we call it. But if it is more than 50 amino acids, then we call it a what? A protein. So now they're saying the DNA molecule consists of 150 amino acids. Don't forget that each amino acid is being coded by 3 anticodons. Yes, 3 bases on the transfer RNA. So they are saying how many nucleotides, and each anticodon, each base on messenger, on transfer, on DNA, each base codes for a nucleotide. So remember, a nucleotide looks like this. Codes for a nucleotide. This is what call it a nucleotide. So now they're saying that how many nucleotides are likely to be present in the DNA molecule? Remember, they said that the DNA molecule code for a protein containing 150 amino acids. You understand? 150 amino acids, and each amino acid is coded by each nucleotide is coded by 1 anticodon, and then each anticodon, remember, each anticodon, if this is a transfer RNA carrying, it has 3 of them. If it is U, A, G, 3 of them, we call this one anticodon. If this is T, R, N, A, yes, and then this is amino acid. So if this is amino acid and this is transfer RNA, then it means that this is going to be the anticodons. Yes. So if this anticodon, the 3 bases on messenger, each base, you see, nitrogenous base, each base codes for an each base code for is like it forms what you call the nucleotide. So now it means that if I have 3 bases here, it means that I have 3 nucleotides. So it means that one amino acid is equal to 3 nucleotides. You understand? So now they're saying we have 150 amino acids. So how many nucleotides are there? So I'm going to say that I have 150 times 3. So it's going to give me 0 here, 0 here, 15 here, 15 here, and then I take 1, 3, which makes them 4. So it's going to be 450 nucleotides. So the answer is going to be B. That's how you can find it there. Sometimes we bring the nucleotides and you ask you how many amino acids are there. Sometimes we bring the bases, the codons, and then we ask you how many transfer RNA are going to be there. So we can bring the question in a different angle. So now we have seen the answers of this. Now let's go to the terminologies. You cannot go to the paper without knowing the terminologies. The building block or the monomers of DNA. The building blocks. Now, next time I'm going to show you the booklet or what we have covered versus what it is here. So the building block of, or the building blocks of the building blocks or the monomers of nucleic acid. How do we call the monomers? The monomers, remember, the monomers, it means that the bricks which make up the DNA. Nucleic acid is messenger RNA and is RNA and DNA. So now the answer here is going to be nucleotide. Nucleotide. Why? These are the nucleotides. Remember, nucleotide, we said it's this, which phosphate, sugar, and nitrogenous base. You are supposed to draw it nicely. Ne? A phosphate, we said, is a phosphate, a nitrogen, a sugar, depending on which, and then a nitrogenous base. So these are the building blocks. So we call this one a nucleotide. Then the gap in the nuclear membrane through which the messenger RNA leaves the nucleus. The gap, the hole. So it becomes the nuclear pore. Yes. So don't forget about that. That's where messenger RNA moves out of the nucleus and then it goes to the ribosome in the cytoplasm. Then the natural shape of DNA. Many students, they will say double-stranded. It's wrong. It's not double-stranded. They are looking for the natural shape of DNA. Is double helix. We call it double helix. That's the answer we want. Nothing else. Double helical, it's not correct. We're supposed to say double helix. Is helical? No. We want double helix. Guys, learn how to give the correct answer without the examiner trying to figure out whether your answer is correct or wrong. Give the correct answer direct. What we ask you, what we give you is what you're supposed. Look at those slides I send you. I send you, or click on the link. You go then look for those slides on our website. Then you'll find out the correct answer. Different color means marking point. Okay. Then they're saying that the sudden change in the nucleotide or sequence of DNA, change in the DNA composition, arrangement of nucleotides. We call it mutation. We call it mutation. But it depends. If it is genes, because they're talking about the sequence of the nucleotide, meaning that those are bases which are going to change. It's only a part of DNA which is going to change, and these parts of DNA, they code for they determine what called a gene. So it means that it's only part of a sequence which changes, and it means that then it is a gene. So it's going to become what you call gene mutation. So change in the genetic composition of an organism, in science we call it mutation, but because it is happening in the gene, then we call it gene mutation. Then when we go to this structure, before you do anything, first identify which structure is that. Yes, first identify and then find out which structure is that. Okay, now here, what we see here is we are seeing DNA. The structure of what? Of DNA. Why am I saying it's DNA? Because they are saying it here. You find out that what is it? Why is it saying DNA? But they said it here. Students tend not to read these instructions which are being given by the what? By the examiner. All right. Identify the molecule. Identify the following. Molecule W. Look for molecule W. What is molecule W? They're saying this. We say that this is a nucleotide. Why are we saying it's a nucleotide? Because we know that the circle means phosphate, and then this one means a pentose, or what you call its name is a ribose. But because it is DNA, then it's going to become deoxyribose. And then this is thymine, which is a nitrogenous base. So a phosphate, a sugar, and a nitrogenous base forms what you call a nucleotide. So W is a nucleotide. What about U? They are looking for the whole structure. What is it? Eh? And then they say that this is DNA replication. Definitely, the whole structure is DNA. Yeah, it's like they're asking you the answer which is already given. And then they're saying that parts of molecule WX, molecule labeled W, part of molecules W labeled X and Y. Where is X? X is a phosphate sugar. And then Y is a deoxyribose sugar. Don't say sugar, we won't give you. Don't say pentose, because we want to know. Pentose means 5-sided sugar, but we want to know which kind of pentose is that. So it's going to become deoxyribose sugar. And then bond Z. Bond Z is weak. You want to see the word weak. Weak hydrogen bonds. Yeah, if you write your hydrogen bonds, your answer will be having some questions, but we prefer you saying weak hydrogen bond. We call that one a KO, meaning that knockout. When you give the answer, no one is going to question about your answer. And then they're saying that where in the cell does this process takes place? Where does DNA replication takes place in the cell? Where does it actually? They have given you the answer because you know that DNA is only found in the nucleus in the cell. If you are talking about DNA, this kind of DNA replication, leave alone the mitochondrial DNA, the chloroplastic DNA. Where is it found? It's found in the nucleus. It's asking you, where does the king found? It's found in the palace. Simple. So it's found in the nucleus. So this process occurs in the nucleus. Yeah, simple. And then they are saying that name the phase of the cell cycle during which replication takes place. Which part of the cell cycle, where do we find this in the cell cycle? That's basically what they name the phase of the cell cycle during which DNA replication takes place. Remember, what is the function of DNA? To double the chromosome number. When does the chromosome number doubles? It doubles before the cell divides. And which phase is it? It is interphase. So it basically, you can say that before the cell divides or during interphase. During interphase, then you'll be able to get the answer here. So interphase. Then explain the consequences if the type of bond Z is not able to break. What if these bonds don't break? What if? Definitely you won't have two strands. Yes, it means that there is no unzipping. Yes, there will be no unzipping. So if there is no unzipping, it means that there will be no formation of new strand, and then it means that there will be no DNA replication. You see, so if it fails, it's like you have failed to open your trouser. What is going to happen? They're going to cut it. If you don't want to cut it, then definitely it means that you remain with your trouser. You can't remove it. So the trouser should unzip first, so that now it can create a space for you to remove it. So if this fails to unzip, yeah, if it fails to unzip, it means that the new strand, this strand which is here, this strand will not form, and also this strand will not form because we will not have a mirror on it. So therefore, we say that DNA replication will not take place because the two strands will not form. There will be no unzipping. Yes, and then it means that the new strands will not form. So basically, that's what you can say. And then lastly, on the DNA molecule, if you bring such a diagram, this is a diagram which shows the protein synthesis. Protein synthesis. So basically, they're saying the diagram below shows the protein synthesis. They have mentioned it. Whatever happens in the nucleus, that is transcription. Yes, transcription. And then what happens in this is the nuclear membrane. What happens in the outside in the nucleus, then is the cytoplasm. Then that is translation. So you have to know this. You have to know that. Then now they're saying that what is X? X, you know, that's DNA. You have to label them maybe before you start. And then what is this? This is a messenger RNA has been formed. And then what is this molecule which looks like that? It's a ribosome. And then basically that's it. And then now you can start answering the question. Identify molecule X. We have seen it. We say that this is what? This is DNA. Ne? We have seen that that's DNA. Organelle Y. What do we call it organelle? Because it's double membrane. Whatever structure is double membrane, we call them organelles. So now, organelle Y is ribosome. The body, zome means the body. Ne? Ribo is coming from the word ribose. Ribose. It is a ribose sugar, so it means that it's a body made up of ribose. That's why we say that the RNA forms the ribosome. Why? Because ribosomal RNA, it forms the ribosome. That's why we call it is a body which is made up of ribose sugar. And remember, this is a ribose. All right. Nitrogenous base 1. Nitrogenous base 1. Where is nitrogenous base 1? Nitrogenous base 1 is here. If this is cytosine, then definitely this is going to be guanine. Then they are saying describe the role of DNA during transcription. Then you define what's transcription, formation of messenger. So DNA forms the messenger, and then this messenger code for specific code for the protein. It carries, the messenger carries the information from DNA for protein synthesis. So basically, DNA forms messenger which carries information for protein synthesis. Yes. So if we don't have DNA, we don't have protein synthesis. The table below shows the amino acids that correspond with the different DNA molecules. They are saying adenine, arginine, methionine, glycine, and then arginine. So they are saying they are giving you. So before you start anything, first identify what is here and what is being given. So write down the correct sequence of amino acid. Now you see, they're looking for amino acid. Sometimes this is messenger, sometimes this is transfer. And sometimes don't say messenger transfer. No, we say codons and anticodons. You have to know that when you say code, we are talking about, you see, we are talking about DNA. We are talking about DNA. When you say codon, we are talking about messenger RNA. When you say anti-codon, we are talking about transfer RNA. You have to know this because we also going to, sometimes we use this language to bring about that. So now write down the correct sequence of amino acid on the structure S. Then we go back to structure S. Where is structure S? Where is structure S? I don't see structure S here. Structure S. What is structure S? Yeah, structure S is this. So now you have, it means that they are saying DNA because here in the table, they are showing you, here in the table, they are showing you code. It means that what we are going to do here, we are going to change this into DNA so that now we can read it here. We can read it from the table. So I have A goes with what? A goes with the T, and then U goes with the A, and then G goes with the C goes with the G, and then again C with the G, and then A goes with the T, and then A goes with the T, and then G goes with the C, and then A goes with T. So now I'm gonna say this is 1, this is 2, and then this is 3. So now I come here, and then I find out which one is 1, which one is 2, which one is 3. ATC. I come here, I look for ATC. So the first one, this one is going to be number 1, and then I have GGT is gonna be, glycine is gonna be number 2, and then I have TCT is gonna be number 3. Then you write them in order, then you get your marks. Guys, I know that this part always disturb student. If you really need more question, or just comment down in the comment. Just comment down that you need more help on this. Then we shall help you, or we shall do more classes concerning about that. Then now we have what we call if the 9th base on molecule S was changed to C. If the 9th base on molecule S was changed to C. Yes. And then explain the consequences, what will form. They're saying 9. So if I have, this is 1, 2, 3, 4, 5, 6, 7, 8, 9. So if this was changed to C, so it means that what I'm going to form T, C, and then this A was changed to C. So it means that A was changed to C. It means that it's gonna have G. So I look for TCG. TCG is arginine. But the third base was this, It was this, it is arginine. Also, this is also coded for arginine. Since it codes for the same amino acid, then it means that there will be no change in the protein, and then this will not change the functioning of the organism. But if this code for another amino acid, then it means that you're going to form a different protein, and even the functioning of the organism is also going to change. But because the one which is being replaced is the same as the one which was original, 3 is the same as the new one, then it means that there will be no change in the protein and they will remain the same. So basically, that's what you are supposed to know. It's like asking you, calling you Karabo, and then the same time you are, for example, you are Kabero, for example. I'm just giving an example. So if I call you Karabo, you say yes sir. If I call you Kabero, you call yes sir. But if you are something else, maybe you are Tapero, and then I call Tapero, then you are not Tapero, then it means that you will not respond. So it means that the functioning will change. If it codes for the same amino acid, however much the base has changed, then we say that the DNA is not going to, the protein is not going to change, and the functioning is not going to change. But if it codes for a different one, then it means that the functioning is going to change. Guys, I love you so much. I'll see you next time. We're going to look at other questions concerning about meiosis. If you have any question, drop it, or WhatsApp us. Then when we go to the general discussion, then we can discuss about it. Thank you very much. I'll see you next time.